Synthetic technologies allow scientists to venture into uncharted waters, asking unique research questions and finding previously unattainable solutions to some of life's biggest mysteries. From gene editing to protein engineering, synthesized DNA libraries enable researchers to grasp once unreachable high-throughput screening applications and dismantle barriers between experimental ideation and execution. In this podcast series, Synthetic Screens, Genes, and De Novo Proteins, the Scientist Creative Services team talks to experts about their experiences implementing Twist Bioscience's synthesized long double-stranded gene pools, called Multiplexed Gene Fragments, for high-throughput screening. In this episode, Niki Spahich from The Scientist spoke with Jeffrey Chang, a graduate student in Nick Polizzi's laboratory at Harvard Medical School and the Dana Farber Cancer Institute, about designing ligand-binding proteins from scratch and testing them in a high-throughput manner using synthetic DNA libraries.
Hi, Jeffrey. Thank you for joining me today. I'm excited to learn more about your work understanding protein ligand binding through de novo protein design. First, tell me how you got into this area of research.
I've always been into science my whole life, and I've been non-discriminate about what areas of science interest me. I find it very curious to learn about the natural world, what's around us, how it works, how we can exploit our knowledge to do useful things for our society. I've had kind of a long, meandering journey so far. I studied physics in my undergrad because I thought it would lend its way to learning more about all the different sciences. In grad school, I ended up in protein design.
What interests you about this field, and how do you go about designing proteins?
There's so many fascinating things that natural proteins can do. After we've spent decades trying to understand them, I think we're at the point now where we have enough data and we have strong enough experimental abilities to make new proteins based on what we know. One of the mantras of our field is that you know you understand something when you can make it yourself. And so after spending so many decades understanding how natural proteins work, our thought is to use that knowledge to make our own artificial ones. De novo protein design is, as the word suggests, to make proteins from scratch. A lot of protein engineering work takes an existing natural protein and makes a couple of mutations or does some directed evolution to get it to be more optimized in certain properties or carry out a slightly different function. Our goal is really to start from a clean slate, start from nothing and design every amino acid in the sequence for a new function. And the purpose of this is twofold. One is to show that you understand the principles of protein structure, protein function, that you could make a new fold from scratch. And the other is that you can imagine there's different things you might want proteins to do, and there's no natural protein that's suitable for it currently. So you really have to start from scratch. With de novo design, you have the power to invent and create something completely new. One other argument, more on physical chemistry grounds, is that native proteins are often marginally stable, and their melting temperatures are kind of low, whereas de novo proteins, due to how they're designed, tend to have very high melting temperatures. They tend to be very stable, which means that they may be more robust to making different mutations to form an active site.
What particular area of protein design do you and the other members of the Polizzi lab focus on? And what are some of the practical applications for this work?
Our lab is trying to focus on ligand binding. Each one of our projects has a different target ligand we're trying to create a binder to. It's a fairly challenging field. Most of us are focused on small molecule ligands compared to peptides or protein-protein interfaces. There's some applications that we were curious about. One of them is metabolite sensing, to be able to detect certain small molecules. There's also thoughts about using this for drug delivery purposes. You can imagine there's certain biomarkers that are present only in a tumor microenvironment. And if you were to somehow design a drug that have two parts, one that's the effector and then one that recognizes specifically that tumor-specific biomarker, it can help target the drug towards the tumor. And some of us are thinking maybe in the chemical industry as well, if we can turn to designed enzymes being catalysts. You can imagine industrial reactions taking place under much gentler conditions that have a lot fewer pollutants, or maybe towards energy methods. Imagine if there's some artificial photosynthesis that we're able to design that could make fuels.
Could you explain how your protein design and test cycle generally works?
There's both a computational and experimental bit. Design cycles where We spend some time modeling things on a computer until we're satisfied enough. And then we order those DNA sequences. Then we express them into proteins and we assay them. In our case, since we're designing ligand-binding proteins, it's various kinds of binding assays. Based on the results, if they bind to the target ligand, then we're happy. We try to analyze it further. So far, this hasn't happened very often. We've only been around for 2 years as a group. And it's also a very challenging thing to do. So what tends to be more common is we try to analyze the negative data, learn from that, and feed that information to the next round of designs.
I understand that protein design is a rapidly evolving field. What advances have you seen recently?
The design tools that we use, a lot of it's changed in the last couple of years. Historically, the field has used a lot of physics-based modeling and classical approaches, Monte Carlo sampling, molecular dynamics, these kind of things. But with AlphaFold and other related deep learning tools, we're now using a whole different suite of tools. We're trying to figure out as a field how to evaluate these new tools, how to fine-tune these models to have a better level of control to get them to output things that are closer to what we want. On the experimental front, a lot of us are doing more large-scale screening with techniques like yeast display, phage display, mRNA display that lets you create libraries of millions to billions of these sequences and then test them all at the same time.
That sounds like a fascinating process. Can you tell me more about the experimental portion? Once you have computationally developed a series of sequences that you would like to test, what happens next?
At the end of the computational phase, we filter our designs by which ones have the best ranking metrics, such as you fold them with AlphaFold and see if they're predicted to fold properly. Then we subselect from those 10,000 sequences to maybe 3,000, and we send Twist a spreadsheet of the sequences. They ship you a tube, but the DNA oligos all pulled together. We take the DNA library and we transform it. In our lab, we're doing yeast display right now. So this gets us a yeast library that has all of the DNA sequences that we ordered from Twist. Then we do yeast display as a way to screen for ligand binding. Your ligands are fluorescently labeled, and you incubate them with the yeast. The yeast themselves are expressing the protein as a fusion to something on the yeast surface. The members of the library that are binding to the ligand are going to be fluorescent because the ligand is fluorescently labeled, and then you can isolate this using a cell sorter and test thousands of different designs all at the same time.
What benefits do the Twist Bioscience oligo pools provide in your research?
The oligo tools by Twist are really amazing for us. They're a way to synthesize tens of thousands of DNA sequences at the same time. It lets us test things in very large scale. And you get more control over the sequences that you test compared to other methods such as error-prone PCR. The power of the oligo pools is that rather than a random library of random sequences, hopefully the sequences that we put in the spreadsheet of the ordered DNA sequences are more likely to work than sequences that are just a random selection. That's the hope of protein design. It lets us beat natural selection and random mutation in a way, because we're focusing our search in an area of sequence space which we think is more likely going to give us functional hits.
I've heard that you're starting to use multiplexed gene fragments in place of shorter synthetic gene oligos. Why make the switch?
So the restrictions of the oligo pools currently is they only go up to 300 base pairs, which roughly corresponds to 85 amino acids. This is a bit of a challenge for us because ligand-binding proteins need to be a bit larger than proteins that just need to fold. The reason is that they have to have a binding site, and if the binding site has polar residues inside, it destabilizes the protein as a whole. It's a hole in the protein that interferes with the overall thermodynamics of the hydrophobic residues being buried. So to compensate for that, the rest of the protein has to have more stability. One way to do that is just to have more of the rest of the protein. And it just kind of gives you more wiggle room to be able to design a larger protein. The multiplexed gene fragment increases the oligo length from 300 to 450 base pairs. So we've been able to design a library of proteins with different folds using that and screen them on the yeast display format. There's one project which involves de novo scaffold design. When we have a target ligand, we have a set of protein folds that we know are designable, and we try to select between them to see which one's most suitable for the ligand. But one of the downsides is that you're not really tailor-making the fold of the protein for a ligand. You're selecting from what's available. And we're curious if we could have a ligand-based scaffold, one that's tailor-made to the specific design problem of a certain ligand target. We've been trying to assemble together smaller structural fragments in a way that both can bind to certain functional groups on the ligand, but also still be able to be assembled together into a full protein scaffold. By the nature of this design process, a lot of the amino acids we end up with are too long to fit in the standard twist oligo pool, but they fit in the 450 base pair multiplexed gene fragment pool. That gave us, with our cloning overhangs, 133 amino acids.
Thank you, Jeffrey, for your time today. I have one final question. What do you think the future holds for your research and the de novo protein design field?
I understand that what we're trying to do is really, really hard. It's taken nature millions of years of evolution and so many mutations and failed attempts to get something. And I think it's kind of presumptuous for us as humans to think we understand proteins well enough. I'm excited to see the success rate go up of attempts we're making. And I'm also excited to see protein design tackle more complicated problems. We view ligand binding as kind of the gateway into all sorts of other things you might want design proteins to do. Once we know how to place a ligand a certain way, design an active site, and then tune the pKa of some residue in a certain way to favor a certain reaction, that would be really cool to see. There's a lot of power in being able to design every single amino acid in the sequence and design so many sequences at the same time to screen them. We're really still figuring out the power of the new tools. Now that we can sequence DNA so cheaply, make libraries so large, I feel like the next 5 years, 10 years, we'll have so much data, and the question is going to be how we're going to use that to not just train new machine learning models that let us design proteins, but also how to fine-tune the models to get them to do exactly what we want.
Thank you for listening to Synthetic Screens, Genes, and De Novo Proteins. And thanks again to Jeffrey Chang, graduate student at Harvard Medical School. Join us for our next episode where we discuss AI-designed binder libraries with Pierce Ogden, co-founder and chief scientific officer at Manifold Biotechnologies. To keep up to date with this podcast, follow The Scientist on Facebook and Twitter, and subscribe to The Scientist Lab Talk wherever you get your podcasts.